Hello, hi, good afternoon. Am I audible?
Yes, ma'am. Good afternoon. You're audible.
Okay. So today we will have very short class because I'm not well.
Yes, ma'am.
So okay. So is my voice clear?
Yes, ma'am. It's clear.
Just a minute. I can hear echo. I'll correct this. Just give me 1 minute.
Yeah, sure.
Okay, let's start. Hello everyone, good afternoon. My name is Kajal Gachawa. I'm PMR Research Scholar from ITV Journalism. I'm your TA for the School for Host-Pathogen Interaction. And today we are at week 11. After that, only 1 week is remaining. So if you have not seen the videos yet, you can check it out from the YouTube channel. And lecture material and the assignment for all the week 12s and the complete course of this host pathogen interaction is already released on the NPTEL channel. So you can check it out there. So let's start today's class. In last class, we have discussed about influenza virus. So would you like to quickly summarize what we have studied about this influenza virus in the last class? Is that clear to you, the classes till now? If you have any doubt from the previous classes, then you can ask. We can start with that.
Yes, ma'am. The influenza virus is spherical to ovoid shape and it is a single-stranded RNA virus with a segmented genome which has 8 different segments and each segment has its own purpose in the infection. And then there are different types of influenza virus. Generally, there are 4 types classified: A, B, C, and D. And A is considered as the most dangerous, and D type infects pigs and cattle, and it does not infect human. Then the nomenclature was also being carried out for based on the different criteria, like the antigenic type, that is A, B, C, or D type, and the host name was also given in the name of the virus, etc. The year as well as the serial number and the geographical location. Then that was the basic thing that we saw in the last class.
Okay.
Ma'am,
Absolutely
also,
correct. Yes,
yes, ma'am. I
Shweta.
wanted again to go through about the antigenic drift and antigenic shift. Could you please do it?
Okay, yes, sure. Just give me 1 minute. I'll open that presentation of last class.
Yes, ma'am.
Okay.
Thank you.
Okay, no problem. It was in just last class or before that?
No, ma'am, it was in the last class.
Okay.
There was a table. You had a little table.
This one?
Yes, ma'am.
Okay. So see, what is antigenic shift? First, we see about antigenic drift. It is on a small parameter. It is a small and gradual change in the like this surface receptors change, or this is very small and gradual change, an accumulation of mutations over time. And after like few years or after a long time, this virus 1 will change into virus 2. Like you can see the initially the receptors were different, and after in the virus 2 the receptors are different. So it is these are the only receptors are changing or the outside things are changing. So it is accumulation of mutations over time. But in the case of this antigenic shift, it is a complete shift. Like see virus 1 and virus 2, two different viruses are combining together. Their viral RNA is combining together and they are forming a complete different type of virus. So this is antigenic shift, like it is completely shifting from there. So you can see from here, That antigenic drift is a gradual alteration in the viral gene caused by mistakes in replication. Like there can be mutation during the replication over a short, like gradual slow changes, like in one replication cycle some mutations are incorporated, in next replication cycle more these mutations are incorporated, that's in that way. And in the end it creates a complete new strain of virus. This is antigenic drift. And in general, the antigenic drift within the genes that code for antigen binding site reduces the effectiveness of antibodies because these changes are occurring on the antigen binding site, like on their surface, these mutations are occurring. And this make the spreading of disease within the host less likely because they are just mutated from that host only. And but in the case of antigenic shift, you can see it is a swift and massive antigenic shift in an infection. Like typically, it is required for two different strains of virus to be combined to create an entirely new subtype that is completely different from both initial virus 1 and virus 2. And a new type of virus has a mix of antigens on the surface of the original strains. Like there will be two different categories of antigens on the surface. But in the case of antigenic drift, only the antigen binding sites were little bit altering, they were little bit changing. But in the case of antigenic shift, complete new antigen binding sites are there, completely new antigens are there. And this can happen because of the gene reassortment that also results in phenotypic changes, like overall receptors are changed. And genetic material is also changed. It is as a result of two different viruses. So these were antigenic shift and antigenic drift.
Yes, ma'am, it's clear. Thank
It's
you.
clear? Any
Any
more
yes, doubt?
doubt?
No, ma'am, no.
Okay. If from any of the previous classes you have any type of doubt, then you can ask. Feel free to ask, no problem.
Yes, ma'am, sure.
Okay. And in the next Saturday, we have last class for this course. So if you have any doubts from the previous classes, you can note it down. But please note with the like week number from which week material you have the doubt so that it is easier to find out. Then we can discuss this in the coming class because that will be your last class for next week.
Yes, ma'am, sure.
Okay. So let's start today's class. This influenza virus structure we have studied in the last class that there it is a segmented, its genome is segmented into 8 different segments and each segment is coding for a different type of protein that have a different type of role. So I will quickly summarize this what we have studied from the last class that influenza virus is a negative sense a single stranded RNA virus. This negative sense means its genetic material cannot be directly translated into proteins. It has to be converted into the positive sense RNA strand first, then it can be translated. And it has a spherical to oval shape. Range is 90 to 100 nanometer, and it has a segmented genome of 8 segments, and its size is 13.6 kb. So it have like some structural and functional proteins, which are mainly the like hemagglutinin and neuraminidase that you might have seen in the last class in detail. And it also have like M1 and M2 proteins. These are responsible for the viral assembly and the uncoating of the virus. And it have neuraminidase, which is responsible for budding of after a newly virus is formed, and it also have this hemagglutinin which is responsible for attachment to the host cell. Then it also has some of the like these nucleoproteins which are NP. So these nucleoproteins are they are responsible for wrapping around the viral DNA and they forms the viral nucleoprotein complex which is essential for its replication and transcription. So there are different type of these proteins. So these we have already discussed in the last class. So after knowing the structure of the virus, next is its mechanism of entry, like how this is getting entry inside the host cell. So influenza virus requires a low pH for initial fusion and therefore internalized by endocytic compartments. And there are four different mechanisms for virus entry. First one is this clathrin coated bits. These clathrins are present on the proteins present on the surface. So the cell membrane forms small bits lined with a protein called the clathrin. So this clathrin have a role in forming a small bit. Then this viral will bind to the receptor and clathrin bits will be formed around this. And then it will be pinched off to form an endosome, like you might have seen in the typical figures of the endocytosis and that all. So it is similar. But the place, like if it is a membrane, here clathrin is coated. So this virus will come here. It want to get entry inside this. It will come and bind here. Then this will be formed here. This clathrin. Then this virus will be inside here virus and this clathrin coated bit will be pinched off and it will get entry of the virus inside the cell. The next one is caveolin mediated entry. These are the small cholesterol rich invaginations like clathrin coated were rich in clathrin protein and this caveolin mediated are rich in cholesterol rich invaginations of the membrane. These viruses use this route to enter via the like flask shaped structures. Like here they are getting into spherical endosomes like structure. In caveoli, they will enter like a flask shaped structure. And this is less commonly used by the influenza, but viruses can enter through this caveoli mediated pathway. Next one is by non-clathrin and non-caveolin mediated. These routes are independent of both of these above mechanisms and this is used by few of the strains for entry. And fourth one is through micropinocytosis. Pinocytosis is like you can say gulping, like if we gulp something inside us, like nigalna type of. So that is pinocytosis of some liquid things. So the cell engulfs large volume of extracellular fluid and sometimes the entire virus inside this. So this is like gulping. Pinocytosis means gulping of something like liquid. Endocytosis of some liquid things. So this membrane will engulf the surrounding extracellular fluid along with the virus. So the virus will get entry. So this is micropinocytosis. And the uncoating of influenza virus in endosomes is blocked by change in pH because pH is required for initial fusion. If we change the pH, then it will not allow to fuse and get entry inside that. And like effective uncoating is also dependent on the presence of this M2 protein, which has ion channel selectivity. So if we modify or alter the ion channels, then this virus will not get entry inside the cell. And there are these two of the drugs which are responsible for inhibiting the replication immediately following virus infection. If virus has infected, then these two will block the replication and they will not allow to replicate. So these were the mechanisms of entry. Next is pathogenesis in humans. So it infects the superficial cells of the respiratory tract and alveolar macrophages and dendritic cells can also be infected. And the site of optimal growth in the respiratory tract for influenza virus is determined by the prevalence of these sugars, that is sialic acid, galactose, and these receptor. So where it will get entry and how it will infect is mainly dependent on the presence of these receptor sugars. So this you can see the surface glycoproteins. Glycoproteins are the sugars. Like these are the proteins which are glycosylated, which contain the sugars onto them. So here in the case of influenza, the most famous example of the sugar counts is sialic acid. Have you heard of this sugar? Have you heard about sialic acid?
No, ma'am, only in the lecture.
Okay, so you are listening this for the first time, so I'll try to explain you this better. So there is a sugar called sialic acid. This here, this purple color triangle you can see here, the small diamond shaped purple color, these are your sialic acids. So the chemical structure of sialic acid is this. It's shown here. and its name is also N-acetylnuraminic acid. It is also known as N-acetylnuraminic acid, which is written as this new AC, nuraminic acid, N-acetylnuraminic acid. So these are the these are family of negatively charged sugars which are located at the terminal end of the glycans on the surface of the cell. See here, this is the surface. Here, there's some receptor or this protein is there. And on proteins here N terminal, the sugars are attached. This sugar moiety completely, this is called glycan because this came here as a process of glycosylation, which is a post-translational modification. Have you heard about glycosylation? The post-translational modification like phosphorylation, glycosylation, that things you have heard before?
Yes, ma'am.
Okay, so because of the glycosylation, these glycans or the sugars are added here onto the surface. And in this case, here the terminal sugar is this purple diamond, which is sialic acid. So these glycans are typically attached to either the glycoproteins or the glycolipids. And this structure of sialic acid or N-acetylraminic acid, it is a 9-carbon backbone which have a different type of sugars. And this structure includes like carboxylic group and this also contain this amino group and OH on its surface. So for in the case of influenza, if we correlate this, then what happens? This virus will use its heme agglutinin protein to bind to the sialic acid residue. Like in the virus, we have seen this heme agglutinin HA, which is responsible for the attachment. It is in virus. So this heme agglutinin will try to bind with the sialic acid. It will try to find the attachment site onto through this sialic acids. And this linkage between this hemagglutinin sugar and the sialic acid determines whether it will get entry inside or not. Because in all the cases it is not able to bind. But if it is able to bind, it will get entry. Because this is a host surface here. This is a cell of the host. Here the host is there. On the host cell surface, there are this protein and the sugar glycans are present. Then from here, this virus will come. Virus have heme agglutinin on its surface. So this heme agglutinin will try to bind with the sialic acid, and if it is bound, then it can get entry inside the cell. So I hope this is clear how the sialic acid are playing a role in virus entry.
Yes, ma'am.
You just have to keep in mind that this heme agglutinin protein is responsible for binding with the sialic acid. That's it. The main key concept is you have to keep in mind. And next is the influenza virus replication cycle. Like there are 4 stages. First one is viral attachment, penetration, and uncoating. In this stage, this is happening at the cell surface, and in this stage, the virus attached to the sialic acid receptors that we have just discussed using its hemagglutinin protein. And after binding, it is getting endocytosis via the glycerin coated bits that we have just seen. And once it get entry, once the virus is inside the host, then the endosomal environment becomes acidic, which triggers the fusion of the viral envelope with the endosome membrane. And this fusion is associated with the M2 ion channel, which will acidify the interior of the virus and it will allow the uncoating. So the first stage was viral attachment. It happens through the hemagglutinin protein of the virus via sialic acid content of the host. The next was penetration. It happens via endocytosis through glycerin coated bits. Next was this uncoating. This happens via the M2 ion channels, which acidifies the interior of the virus. So these things you got from the virus structure. This is because of hemagglutinin. Penetration is via the uncoated bit, and this uncoating is via M2 ion channel. This will acidify the interior so that it can get uncoated. Once the viral assembly is uncoated, the next is its work is to do the transcription and translation. So, as we have seen that influenza virus is a negative sense RNA virus, so its genome cannot be directly translated into proteins. And this virus have its own RNA dependent RNA polymerase complex. This enzyme will transcribe the negative sense into the positive sense mRNA. Then this can replicate inside the nucleus. So the second stage is transcription and translation. Then the third is viral RNA replication. Like it carry its own polymerase complex, so it can get replication also. Once it is replicated, the next stage is to get matured, like maturation, so that it can form multiple particles and it can bud out of the cell and can infect the nearby other cells also. So at maturation stage, what will happen? The viral nucleoprotein complex, these are exported from the nucleus with the help of this nuclear export protein. NEP. These are what? Nuclear export protein. NEP. So what will happen? These viral structural proteins like hemagglutinin and neuraminidase are sent to the host cell membrane. And the viral component assembly, it will assemble at the membrane and then this virus will get bud off from the cell. Like as it got endocytosed by forming an inside bud, similarly it will do the exocytosis and it will get outside. So you are getting some like how these proteins that we have discussed that it contains, how they are responsible from here. Like for first attachment, the hemagglutinin is there. Then for uncoating, the M2 ion channels are there, which will acidify the inner conditions and all. Then for viral assembly, then for replication and this transcription, it have its own this polymerase complex complete. This will allow it to replicate, transcribe, and everything. After that, for the virus assembly again, just have this M1 protein. And once it is assembled, then for again budding off, it also have this other proteins there for maturation and coming out of that cell. So you got this that how they are doing this.
Yes, ma'am.
And here this is the role of hemagglutinin, which is responsible for attaching to the sialic acid. The other one was neuraminidase, NA. Neuraminidase is responsible for cleavage of the sialic acid. The sialic acid is bound to other sugars like this galactose and this N-acetylglucosamine and this mannose, like as you can see here in the structure. So the sialic acid is bound to the galactose here. So for cleaving this bond from here, like for cleavage, neuraminidase is responsible. And why we require this? Because for getting outside of the virus or budding out from the cell, we don't need the virus to have the sialic acid exposed on its terminal ends. Otherwise, what will happen? The same virus, like this is cell 1, this is cell 2. If this virus is getting outside from this cell, but if it contains the sialic acid on its surface, then it can again reattach to the same cell because this cell already contains the here this proteins are there, so this can get here attachment. So for making it a new virus particle, what will happen? The neuraminidase enzyme will cleave off the sialic acid from here, so that this virus is a free virus. It don't have exposed sialic acids. So it can go and again bind to the here cell to sialic acid using its own hemagglutinin. So there is a role of this again. This neuraminidase, it is again responsible for virus budding off. Like when it comes out, then this neuraminidase is responsible for cleaving this terminal sugar, the sialic acid from here. So at maturation, neraminic acid will play its role. And this NEP will also play its role in nuclear export proteins. And for these two mechanisms, polymerase complex will play a role because it contains its own RNA polymerase and other proteins required for the replication and translation. So, from the structure, can you correlate this to how this is getting attachment, replication, and again budding off? So, this is the four steps.
Yes, ma'am, it's clear.
Okay. And this is the same thing that we have just discussed in like back slides, like how this is getting here inside the budding. This is clathrid coated bit. It is getting inside. Then it got bud off in the inside after endocytosis. Then your M2 ion channels came into role and the uncoating happened here. Then after this uncoating, this transcription, replication and synthesis of viral nucleoproteins happened here. Then again it was packaged into a viral assembly and this assembly attached to here on the cell surface. Then this formed here a bud and it released outside here again. So this is the simple mechanism that we have just discussed in the previous slide. Next is cold and flu. Very common terms that we use in our daily language, our daily life, daily term. I got cold, I got flu. We use these both the terms at same place. We are not aware of what is exact difference. So after going through the lecture material, are you able to differentiate these both the terms? What is difference between cold and flu or are the same?
Yes, ma'am. Generally, most of the common cold is caused by the group of viruses called as rhinoviruses, but most of their symptoms are similar.
Hmm.
So people usually get confused between all the both the flu and cold.
Hmm.
Yeah.
So what happens here in the case of cold and flu? Like this, there are two different terms, cold and flu. The common cold is mostly commonly caused by the rhinoviruses, 50% of the time. And other viruses are responsible for other cold also. But in contrast, the influenza is it is mainly responsible for this flu. Like there are two strains, influenza A and influenza B. These are responsible for causing the flu. Like it can infect your upper and lower respiratory tract and it can cause a severe illness and other symptoms like body ache, fever, and other things. So these are different, these are not the same. Like this flu, it is a sudden onset. Like suddenly in the morning you are perfectly fine, but in the afternoon or night, suddenly you got a high fever and body ache and extremely fatigue. So this is a symptom of your flu. But in the case of cold, it usually develops like gradually and initially it is milder. Like sometimes you feel this, yes, you are not well, you are getting some cold, you have some stiffened nose or some body pain. Then after 1 or 2 days you get high fever. So that is your cold. But flu is sudden. Morning you are fine, evening you have a high fever. So that is your flu. In short, you can keep in mind like this. This your flu is your sudden and your cold is your gradual. Slowly, slowly you will fall ill. Slowly, slowly you will have uneasiness in your body. So that is your cold. And these are caused by different type of viruses. Like in the case of cold, it is 50% time by rhinovirus, and in the case of this flu, it is majorly called by the influenza virus. So these are the like clinical symptoms and all. I think you might have gone through the slides. These are easier to understand. Or if you have any doubt or if it is not very much clear to you, then let me know. I will explain this. Otherwise, I'll skip this.
No, you can skip it, no problem.
Okay. Because I'm trying to convey you the basic mechanisms or the basic principles of how these things are happening, and rest of the theoretical parts that you can read by yourself also, or just listening to the video lecture, you can understand. If you have any doubts from anything, you can let me
No, no, it's okay. We can directly skip to the questions
know.
if
Okay.
no problem.
Okay, I'll take few topics in between,
Yeah, sure.
otherwise we can directly skip to the
Yeah,
questions.
sure. Yes.
Like we have seen the influenza virus as a first type of virus, then next topic is your arbovirus. So, what you understood about this? Like, what is this arbovirus?
The viruses are the viruses that usually spread through insects or arthropods.
Hmm.
And there are many, most of the uh, vectors are mosquitoes.
Okay.
And there are few mites and ticks that also ticks, fleas, bugs, and some flies and lice. They also act as a vector in order to carry the pathogen from one host to the other.
Yes, absolutely fine. So, arbovirus is your arthropod-borne virus, or mediated by these arthropods, or they require any intermediate host, like intermediate vehicle for transfer from one host to other host, like these your mosquitoes, your ticks, and your flies. They are required for transmission from one person to other person. So these mosquitoes can spread like the so many different type of viral infection from diseased person to healthy person. As we are aware of this, like chikungunya and dengue, these are the like dengue and chikungunya are very common on every layman's term, that these are spread by the help of mosquito. Mosquito is not responsible for causing this dengue and chikungunya. They can transfer from one host to other host. And the Zika virus is also spread by the mosquito. Yellow fever is also spread by these mosquitoes, and West Nile virus is also there. So these like arbovirus disease are the transferred by any these intermediate host or vehicles. The ticks are also responsible for transmitting this virus from one host to other. And why mosquitoes come to our body, like why we are attracted towards the mosquitoes? Because of our body odor or our skin bacteria, and also our body temperature. So these mosquitoes find it very favorable and very suitable on our skin, so they come and sit on our skin, and then they penetrate the virus inside your skin, and they cause the infection. So these were your arbovirus. Next one is your dengue virus. Dengue virus is a family of a flavivirus genus. Like there are different families of virus like a flaviviridae, and there are different genuses of this viruses like flavivirus. So dengue is one of them. And this dengue virus is also spread via these mosquitoes like Aedes. Mosquitoes responsible for transmitting infection from one person to another. This is the like transmission of dengue virus. Initially, in our very initial lectures, I remember we have studied about some disease that was being transmitted by some of the vehicle, where we have studied like half of the life cycle of that particular disease is carried out inside the human and half is carried out inside the that vehicle. I think if you have gone through the slides, then you might remember that name. What was that disease? So this virus is maintained by human to mosquito to human cycle. Like human, something will be there in the human, then it will go to mosquito, then it will again go to human. So mosquito is here intermediate. It is responsible for spreading that disease. These are the different stages of this dengue virus. Like how much fever you will get, at what condition, what will be other symptoms with the fever. So these are different stages of the dengue fever. This is the life cycle of dengue virus. I hope is this clear. I think this is very easier to understand. Or if you have any doubt,
No, no, that's clear.
okay, then I'll skip this portion. The next one is your Zika virus. This is a positive sense RNA virus, and the family is same the flavivirus or the flaviviridae family. Then next comes your bacterial infection. You have seen your viral infection, different type of viruses. What are their structures? What is their mode of pathogenesis? What are their type of disease they cause? What are the symptoms they occur? And the next one is a bacterial infection. And in the last week material, I think there is your parasite and fungi mediated something infection is there. So these are the different types of infection. So bacterial infection, this can be like your intracellular like MTB, Mycobacterium tuberculosis, Listeria, or it can be your extracellular like Salmonella and Shigella. And the route of entry can be through your broken skin, any you have any skin damage or any scratch on your skin, then your this Clostridium tetani can enter inside and it causes the tetanus. That's why if we get hurt from any iron material or any sand outside our home, then initially we say go and have a tetanus injection, because if we get any scratch in our skin, it is very easy for Clostridium tetani to get inside. And this tetanus is very much like painful if it happens. Then immediately we are suggested to take the tetanus injection so that if we are exposed to Clostridium tetani, then it don't get spread in our body. Then the next route of entry is via your respiratory tract. This is mediated by your like Mycobacterium tuberculosis. So that's why we say that you should keep a little bit distance from the TB patients, because TB patients have a respiratory disease. They will continuously they will sneeze or they will cough because their respiratory tract is completely infected. So during their coughing and sneezing, the droplet are spread in the surrounding environment, and these droplets contain your this MTB bacteria there, and it can be spread to the if we any healthy person touch those droplets, like accidentally our hand can go there, our clothes can touch that, then that can be transmitted inside us. Like if we have touch our hand on infected surface, then we touch to our own nose or mouth, and that can get inside us. So this is respiratory mediated. That's why it is advised to like keep some distance from the MTB patients. Directly by touching their skin, MTB will not spread. But they have already coughed in that environment. Their MTB particles are already outside in that environment, and that can be touched to our hand or our mouth. The next one is your GI tract mediated, like gastrointestinal tract infections. These are your salmonella and shigella. Like it is advised that in the rainy season you should not eat the outside liquid foods like your pani puri and your juices, because these can contain your salmonella and shigella, because these are waterborne diseases, like mediated transferred via contaminated water, and these can infect your gastrointestinal tract. After that you will have vomiting, diarrhea, and those kind of things. Then the next route of entry is your genitourinary tract. Like we say, somebody got UTI, urinary tract infection. So that can be mediated by this Neisseria gonorrhoeae, which can cause the gonorrhea disease. So that's why it is advised to have some hygiene from the public places. So these are different routes of entry via skin, via your respiratory tract, via GI tract, or via urinary tract. And the infection to the host and disease outcome depends on your inoculum size, like how much you are exposed to that. Like if you have just a little bit of bacterial touch to your surface, it might not harm you. But if you are sitting completely adjacent to the infected person, then you might get sick. So inoculum size matters, like how much you are exposed to that. Next is the virulence of bacteria. If your bacteria is less toxic, then you might not get infection. But if it is very toxic, then even a single exposure can cause you ill. And next is our host immunity. How much strong the immune system of our host. So these are the factors which depends on the outcome of a disease, like inoculum size, virulence of bacteria, and host immunity. These are like immunity against bacterial infection. We have already studied this so many times about the innate immunity and the adaptive immunity, that how our body is responding to that. Next is our infection process, like attachment or adherence to host cells or epithelial cells and multiply. Like first it will get attached to the host surface, then it will get multiplied, then it will spread to other things. And these are the like genomics and bacterial pathogenicity. So I think you might have gone through this. Or is it easy to understand, or is this difficult? If it is difficult, then
No,
let
no,
me
it's
know.
easy.
Okay,
We
okay.
can
If
skip
it's
this.
okay, okay. Thank you. Then these are some examples of virulence factors which are encoded by genes on mobile genetic elements, like how this can get in, like what type of genus or species the plasmid encoded, like E. coli. It have virulence factor is heat labile, heat stable, enterotoxin, endotoxin. These things are there. So by this, the bacterial infection is also finished. So if you have any doubt, then we can quickly discuss, or we'll start the questions and finish the session.
We can start with the questions, no problem.
Okay. So the first question was, which of the following infections is not transmitted via mosquitoes?
Oh, Asin
This
virus because it's transferred by the mite.
this person virus is transmitted by,
Mite.
okay, and other all are transmitted by your mosquitoes.
Mosquitoes.
Okay. So this your person viruses transmitted by you. This is transmitted by your tick bite. Another all are your virus infections. The next question was, which is the mechanism by which secondary dengue infection can lead to more severe disease, such as the dengue hemorrhagic fever? By which mechanism?
Antibody dependent enhancement.
Because in the secondary dengue infections, the immune response is influenced by pre existing antibodies from a previous infection. When the body is exposed to a different strain of the dengue virus, these antibodies can enhance the viral entry into the host cell, particularly through the Fc receptors. And this will result in an increased viral load and more severe disease, such as the dengue hemorrhagic fever and dengue shock syndrome. So, this is mediated by your antibody dependent enhancement. So, this dengue hemorrhagic fever can happen on your secondary dengue infection. The third one is which of the following mechanisms can potentially be targeted to prevent your influenza virus replication?
Inhibition of viral attachment to the host cell, blocking the transcription and translation of viral protein, preventing viral uncoating with host cell, interfering with RNA replication of the virus, inhibiting neuraminidase activity. We cannot disrupt the host sialic acid synthesis, and we cannot also increase the host receptor expression and promote cytokine storm induction.
Okay. So, in this question, the complete mechanism of influenza virus infection that we have discussed, like right beginning from the attachment, we can either inhibit the viral attachment, but if it got attached, then we can block the transcription and translation of the viral protein so that it don't get replicated into more virus particles. Then, if it got replication like transcription, then what we can do, we can prevent the viral uncoating within the host cell. And if it is also there, then we can also interfere with the RNA replication so that it is not replicated. And even if this, then we can inhibit the neuraminidase activity so that at last stage during the maturation, neuraminidase was required for cleaving of your sialic acid. So that we can also do. But disruption of oxalic acid synthesis, that is not in our control. And increasing host receptor expression, it is not required. Either we can decrease the host receptor expression, but that is also not possible. And promoting cytokine storm induction, there is no role of cytokines here in this case. So the correct options are these. The fourth question was what type of virus is your Zika virus classified as?
Positive and single stranded RNA.
Yes. So this is your positive and single-stranded virus. This is your Zika virus. And what type of virus was your influenza virus?
It is a negative and
It
single
is
stranded
negative
RNA.
and single-stranded
Yeah.
RNA virus. Okay. Is this clear to you? What are these single-stranded, double-stranded, negative, positive? Is this clear
Yes,
to
ma'am.
you?
Yes,
Okay.
ma'am.
Okay. So the next question was which virus family predominantly include arbovirus responsible for encephalitis?
Togaviridae.
This is your toga variety. And this family, like the toga variety family, predominantly includes your arboviruses, which are your orthopogon viruses, and they are responsible for encephalitis. Next question was which of the following is not a function of the hemagglutinin protein of the influenza virus?
Viral genome replication.
Viral genome replication, which is not a function of the hemagglutinin
Yes.
proteins of influenza virus. So, binding to sialic acids on the host cell is its main function. And membrane fusion is also its role of hemagglutinin protein only. Then antigenic recognition, yes, only after the antigen recognition it can be able to bind or attach to the surface. And triggering low pH condition, yeah, outside the pH is already low there. So the viral genome replication, this is not a function of hemagglutinin protein. And the next question was why is low pH important for the influenza virus during the fusion process?
Yeah, it's necessary for the entry of the virus into the host cell.
Yes, they are asking about low pH. for the influenza virus during the fusion process. Sorry, just so your low pH is required during your influenza virus entry process because it will trigger the conformational changes in the hemagglutinin protein, and this change will facilitate the fusion of the viral envelope with the host cell membrane. And the acidic environment inside the endosome after the virus has been internalized into the endosome, it will cause this change and it will allow the viral RNA to be released into the host cell and start its replication. So this low pH is required for the successful entry of the virus into the host cell. Next question was, which drug have mechanism that can prevent viral uncoating and replication in the influenza virus?
Chloroquine and amantadine.
This is your chloroquine and your amantadine. So these are responsible for what they do. They target your M2 ion channels. Like in the initial slides, you might have seen that. This here ion channels. We have talked about this ionophores and M2 ion channels for the uncoating. These are responsible for interfering with those ion channels. So it will prevent the viral uncoating and replication. The next question is, which of the following statements is true regarding the influenza virus genome?
It is segmented. It has 8 segments.
Yes, it is segmented into 8 RNA segments, and each segment codes for a different protein that is responsible for the overall pathogenesis of your influenza virus. Then the last question was, which of the following statements about dengue are true?
So, dengue fever is caused by virus transmitted exclusively by mosquitoes. It is Aedes aegypti and Viravan. Then, antibody dependent enhancement can lead to severe manifestation during secondary infection, which we have seen recently. And early detection methods, including RT-PCR and serological antibody tests, can be done. Then, supportive care is the mainstay of the treatment for dengue because no specific drug for the control of the virus is not there, but the other symptoms can be managed with the help of drugs. Then community awareness plays a vital role in dengue prevention and control because you can destroy the breeding places of the mosquito.
Okay. And what about the incorrect options?
No, infection with one serotype does not guarantee the infection with the other serotype, but it will enhance the infection. Like, if we are getting infected by dengue again, it will be more virulent.
Hmm.
Then there are currently no effective vaccines available for dengue. Of course, there are few vaccines available in the market. Then raising global temperature have no impact on dengue transmission. It is wrong. It does have an impact because these mosquitoes are easily found in the regions that are near the equator.
Yes. So these are about the dengue virus infection. So with these questions, we here ends the week 10 assignment, and the lecture material for week 11 is over from my side here. If you have any doubt from these questions, like all of 10 questions are clear to you, or in any question you have doubt, you want me to repeat?
No, ma'am, it's clear.
Okay, so if you have any more doubt, you can ask, or we can continue our session in the next Saturday class.
Yes, ma'am, sure. Take care of your health. Thank you so much.
Okay. Okay. Thank you, Shweta. Thank you for joining. If you have anything, then you can drop me a mail or message.
Yes, ma'am, I'll let you know. Thank you so
Okay.
much.
Okay. Thank you. You can log out.
Yes, ma'am.
Okay.